Hello, my friends. Welcome to Origins. I'm Don Chapman, and I'm your host. It's great to have you with us today. Origins is a forum where we take the evidence of science and use it to confirm the truth of God's creation. We usually do that by bringing in some guests who are experts in the various fields of science and apologetics and have them share with us the truth that God revealed to them. We've got a great guest with us today, Dr. Jerry Bergman. The thing I love about Dr. Jerry is that he is a lifetime student. He told me he has 9 different degrees from the college level up, including several PhDs. He's been a student all his life. You know, learning's not something you do to get a degree. Learning's something you do because you want to pursue the truth. And that's what Dr. Jerry's done all his life. Dr. Jerry Bergman, welcome to Origins.
Thank you.
It's great to have you here today.
Good to
We're going
be
to talk
here.
about mutations today.
Right.
We've been joking about this third ear that I have under my arm and how I grew it through mutation. Is that what this is about?
Well, it's about actually the harm that mutations cause.
Okay. Now, mutations, when we think about it, you can't go very far into talking about serious macroevolution without talking about mutation as the vehicle. Is that true?
Yeah, the basis of evolution is mutations. That's what evolution is based upon. That provides the raw material for natural selection to select from, according to Darwinists. And I've got a couple quotes which I think are very telling about what Darwinists believe and how important mutations are. If I can
Let's do it.
go up here. And as I mentioned here, the primary basis of macroevolution, and macroevolution is what we're talking about here, and macroevolution is evolution beyond the species level, from, as they say, from the goo to you by way of the zoo. That's macroevolution. And mutations are basically variations in the code that produces you and I. And the code, of course, is well known as the DNA code we see over here. This is a model of DNA, and the DNA code is made out of letters, and these letters can change. And the changes are what Darwinists postulate produce the variety which natural selection can select from. So they change the protein coding DNA. That affects the phenotype.
You're saying that the DNA chain is made up of letters.
Right.
It's not really made up of letters. It's made up of chemicals, but it's almost a chemical language, isn't it?
Right. It's made out of chemicals, and we just, instead of saying the chemical, because they're long names, so we just
assign them letters.
Assign them letters, right? We just T and A and G and C.
And so there's really a language written in chemicals.
Right.
Okay, that's great.
True. And of course, there are other sources of variation. We're not saying mutations are the only source. Sexual reproduction, for example, you have 10 kids, they're all different. That's a source of variation. Crossing over, genetic crossing over, is literally where the chromosomes exchange parts before they produce a human being. And this exchange basically allows new traits to appear because the combination changes because of this crossing over. When I explain this to my students, it's like you lay down next to someone and your legs are literally cut off in exchange. So you end up with their leg and they end up with your leg. So you have a difference because of this exchange. And this is crossing over. Transposition is another source of variety, and transposition is literally where the genes cut themselves out and go somewhere else. And therefore they change the code, and therefore they change the pattern, and therefore they change the phenotype, the traits that result. So there are other ways of producing variety. But all this does is rearrange the existing information. You don't have any new information. You basically have information that's there which is shuffled around. A good example would be you rearrange the furniture in your house. You have, looks different, but of course there's nothing new. And
I think it's really important that we camp there just a minute, because if evolution is going to work, there has to be new information.
Right.
And as you said, it doesn't matter how many different ways I arrange the furniture in my living room, we're still going to have that old couch somewhere, and there's never going to be a new couch.
Exactly.
And that's exactly what you're saying about the DNA. There are ways that God has made for there to be variety within that species or that kind, but there's no way to have a new kind in there, is there?
Exactly, and that's the problem. And
Thank you, Fred. That's great.
They recognize that, Darwinists do. That's why Ernst Mayr, who's a picture up here, he just turned 100, by the way, recently. And he said, ultimately, all variation is due to mutation. And this is not a minor figure in Darwinism. He's one of the leading, if not the leading, evolutionary biologists in the world today.
All their eggs are in the mutation basket.
Right.
Okay.
That's critical. And
all right.
Theodosius Dobzhansky, likewise one of the leading evolutionists today, said the process of mutation is the only source, he stresses, only source of the raw material and hence of evolution.
So if we can show that mutation doesn't make evolution happen, the evolutionists themselves would say then evolution can't happen.
Right,
All right.
exactly. That's why this is such a critical area.
All right.
And a good illustration of this is Dawkins, Richard Dawkins, who's also one of the leading evolutionists today. He lives in Great Britain. He teaches at a major university there. And he developed a computer simulation where essentially he took this sentence, "Me thinks it is like a weasel," and this is taken from Shakespeare, and he started out with random letters. These are all random letters. You just generate randomly. And then he developed a computer program that would change these letters one at a time. And the changes, if they met this goal, they were saved. For example, the first here is a W, which of course is not the first letter here. It's an M. So here we have a mutation which produces an M, and then the M therefore is saved, and you can see from that point on the M is retained in the program. And eventually, if we keep doing that, eventually we get to the sentence which is "Me thinks it is like a weasel." And that's an illustration Dawkins uses to illustrate basically how Darwinism works, how evolution works.
You know, I'm hearing you say that, and I understand, I think I understand at least how that happened, but with a computer, there is a source of these letters coming in. A man is not a computer. We don't have an influx of new letters, so is that a valid analogy?
Well, there are a lot of problems with it, and of course, creationists like to use it because it's a good illustration of the problems of Darwinian idea. Dawkins uses it to show how Darwin works, but we use it to show how it cannot work. Okay. Among the problems that exist with mutations as a source of variation are we assume mutations are random, and of course, that's a major assumption. And as we'll show, the problem is we know they're not random. When random mutations occur, certain amino acids are far more likely to be produced than others. So we'll assume for a minute they are random. So we'll just take that as an assumption. But we're going to end up with certain amino acids are far more likely to be produced than others. And of course, these letters, these A's, T's, and so on, they produce amino acids. Amino acids are assembled to produce protein, and protein are the building blocks of you. You and I are made out of protein. And therefore, we get the protein by getting these amino acids. So to produce the protein, to produce you and I, we have to have the right order of amino acids. So it's critical to get the right order of amino acids. And random combinations, I point out here, are more apt to produce certain amino acids. And I'll show you from the following table why that's true. For example, this combination, TCT, here, you can see that plus a TCC and TCA and so on. There's 6 different combinations, all which produce serine. Okay? So we have 6 different combinations. So if we randomly shuffle the 4 letters, we will get 9.4% of the shufflings will be serine. Okay, now we'll go over here and we'll look at tryptophan. There's only one combination of amino acids that produces tryptophan. Therefore, only 1.6% of all combinations will produce tryptophan.
So you have almost 9 times as much chance of getting that first result as the second one.
Right. So we'll end up with a lot of serine and not very much tryptophan. Well, that's a problem because we need actually a lot of tryptophan in many different amino acid combinations to produce a protein. And as a result, tryptophan is very important. And by natural means, we're not going to get nearly enough of this, and we'll get way too much of the serine. So we don't get the right combination we need to produce an organism. It's off balance, assuming now they're random. Okay, a basic problem is that the rate of mutations from one base to another base is not equal. So in other words, they're not random. We don't end up with a random change of letters. And one study which I looked at found the totals, which refers to all base pair conversions, as follows. I put that in a chart to make it easier to follow. And this is the conversion to thymine. So the conversion of this G to a T, C to a T, A to a T, we see is a total of 58% of the total of all conversions. So what that means is that all conversions, we're talking about mutations. We call that a conversion. So mutations, basically, what's going to happen is we're going to end up with an awful lot of thymine because all mutation types are not equal. So we'll end up with a huge amount. Over half of the mutations are going to end up with thymine. Guanine, on the other hand, only 14%. 50% of the total. And cytosine, only 23% of the total. And adenine, only 5% of the total. So applying this now to Dawkins' illustration, you can see what's going to happen is the conversion of thymine is fully 10 times more common than conversion to adenine. And as a result, this is what we'll get. Instead of getting our me thinks it is like a weasel, what we're going to get is a lot of T's, because the T is much more common. Now, of course, some of the T's will convert back to the other letters, but by and large, the T's are going to dominate, so eventually what we're going to get is lots of T's. So we will never, just looking at this fact alone, we'll never end up with me thinks it is like a weasel. We're going to end up with dominance by far of T's.
Now, you used a very dogmatic word there. You said we will never end up with me thinks it is a weasel. Are you saying that it's improbable, or are you saying it's impossible?
Well, it's improbable to the point where we might as well say it's impossible.
The chances become so infinitesimal that it simply couldn't happen.
Right. And there is a very, very low probability, but the probability is so low that we say for all realistic purposes,
realistically, it couldn't happen.
It's basically
so you're
zero.
basically saying that for mutation to be possible, or for mutation to be positive, really isn't possible.
Right, because of this, and there are several other factors too, but this is one factor.
Well, that's interesting. We've got to take a break right now. We're going to talk about hot spots when we get back, so don't go away. This is going to be hot, and we want you to be with us for it. We'll be right back.
Creation versus evolution, you weigh the evidence. Tadpole to toad, self-destruct DNA. Biologists have found that certain living cells come with a built-in self-destruct mechanism. For example, as a tadpole turns into a toad, it no longer needs its tail. When the special gene gives the order, the tail cells begin to die. In other words, some living cells contain a gene that signal the death of the cell at an appointed time. Why would evolution develop genes that order their own death? By definition, such a gene would not aid survival. Today's guest on Origins, Dr. Jerry Bergman, has been a featured speaker on many college campuses and at professional meetings throughout the United States and Europe. His research has been featured in many publications across the world. Dr. Bergman is also a professor at Northwest State Community College in the state of Ohio, where he has taught biochemistry, anthropology, geology, and microbiology. For more information about our guest, you can write to Dr. Jerry Bergman, Northwest State Community College, 22600 State Route 34, Archbold, Ohio, 43502.
We're back, and welcome back. I'm here with Dr. Jerry Bergman. We're talking about mutations. You know, mutations have to be the vehicle through which evolution works if it's going to work. But Dr. Jerry Bergman is telling me that, in fact, mutations prove that evolution can't work. Now, Dr. Jerry, help us first of all. What's a mutation?
Okay, mutation is a change in the DNA code. This is a model of the double helix you can see here, and you've got two helical circles around here, and inside there you can see those bar-like materials. These are what they call base pairs because they're chemically bases, and these base pairs match up together. We have, for example, T matches up with A and C matches up with G, and these two match up together to produce the code, and the code basically is what produces the amino acid set which produces protein, which produces you and I. And every now and then, for a variety of reasons, a mistake is made. For example, cosmic ray may hit this and damage one of the rungs, has to be repaired. And it's usually repaired properly, so we're okay. And we have about a dozen repair processes which are constantly monitoring this and repairing it. It's constantly being repaired. This is going on 24 hours a day. Every now and then it doesn't repair it properly. It puts the wrong base pair in there, and so instead of having a C, we end up with a T. So that's a mutation, so we have a change in the DNA pattern. And these are called mutations.
And now for evolution to work in this modern day and age, Obviously, when evolution started, they didn't know about DNA.
No, didn't have any idea of any of this.
And so to take that 19th century theory and to see it work in the 21st century, somehow the whole business of DNA has to be included in their thought.
Right, and that actually is where scientists don't accept Darwinism now, they accept neo-Darwinism. And neo-Darwinism is where we've added the information relative to mutations. And they thought this would be a savior for Darwinism, but it's turned out to be the opposite. It's turned out to be lethal for Darwinism.
And one of the big kahunas in the neo-Darwin movement is a guy named Richard Dawkins.
Right.
And Dawkins has written a book showing how DNA could work to produce the mutations needed to bring about evolution.
Right.
And you're saying that it isn't working.
Right. It's from what we know about biochemistry and molecular biology, we understand now it doesn't work. It cannot work.
Now we're ready to talk about hotspots.
Okay. Hotspots are, the mutations tend to occur in certain areas. They're much more common in certain areas. In fact, P53, which is a gene we've looked at more so than many other genes, we find that about 70% of all mutations are only in 3 or 4 areas along the entire gene. And some of these genes could be thousands of base pairs, so many are huge. So there's a lot of areas where a mutation can occur. And if the vast majority occur in a very small area, we know there's something going on. And we understand now some of the reasons.
Do we know why there's hotspots?
Yeah, we understand some of the reasons. It partially has to do with the construction of the double helix, where they're located, because some parts of the double helix are more exposed to radiation or other sorts of mutations than others. Certain parts, like when you have a lot of letters that repeat each other over and over, like you have a lot of T's or a lot of G's, that area tends to be prone to a hotspot. Some we're not aware of exactly why there are hotspots, but we know there are hotspots. And there are also cold spots. Cold spots are where they rarely occur for various reasons. And one of the most common mutational hotspots is at the CG dinucleotide. And they're involved in mutations in about 12 times more often than other dinucleotide sequences. So this is one example of many. And there are several reasons why this CG tends to be a hotspot, but here I was just trying to show you that there are a number that we know that exist. In fact, in one study, only 2 mutations accounted for 94.4% of all mutations out of 319 identified in one gene. And there are many more that are possible in this gene. There are maybe 1,000 that are possible. But there are 319 they identified, and then out of these only
almost all of them was at this 2 specific places.
2 places, right. Yeah. And so what does this hotspot have to do with Dawkins' illustration? Well, this shows it. Result of hotspot and back mutation. Now I've combined these together because we have hotspots, and of course hotspots means that you're more likely to have a mutation in that area, but it's more likely to go forward, to change to a T, for example, but also more likely to go back. So this is why we can occasionally have mutations which repair the damage. We can actually have a mutation and it heals the damage. So we can have, in this situation, a beneficial mutation. So if we take the example again, methinks is like a weasel and you go forward. Of course, what we're looking for now is the M, in this case the W actually, to change to an M. Okay, and you can see that's not a hotspot, so that never changes. On the other hand, S happens to be a hotspot area, and the T you can see now is what we start out with. So the T changes to a Y, to a T, to a T, to a T, and we end up, because this is a hotspot area, we end up with accumulation of Ts. So what happens when we look at both hotspots and back mutations, plus the tendency to go from a C and a G to a T, we end up with constant changes in a small area of the code here, so we end up with never getting what we want, but we end up with constant changes to a T in the hotspot area.
So those cold spots, they're never going to change. And the hot spots, you're going to keep changing there.
Or the cold spots will rarely change, that's true. And the hot spots will commonly over and over change.
So Dawkins is assuming a uniformity across the letters, and it just isn't there.
Right. For his model to work, you have to have the uniformity.
And that's not true in the DNA code.
Right.
All right. I'm with you.
And the other thing we need to look at is, you can't only look at base pairs, but you also have to look at polarity, because bases could be polar or nonpolar or semipolar. Because basically these amino acid sets are held together by the polarity. That's a critical aspect in bonding the protein to produce a workable, functional structure. And when we look at these charge differences, we can see that the nonpolar uncharged just by random changes, we end up with 34% of the amino acid sets are nonpolar uncharged. 28% are polar uncharged. 11% are polar, nonpolar borderline. 12% are polar positively charged, and 6.2% are polar negatively charged. Now what that means is we're not going to, by random chances, we're not going to get the combination we need to produce the protein structure we have to have. And so purely by random changes is not going to work. Now hotspots even complicates this further.
It is complicated. Give me a couple of examples of mutations that you've seen.
Well, my work is in cancer research, and doing cancer research, of course, what we're looking at basically are mutations. Cancer is a result of mutations. That's what causes cancer. Cancer is a genetic disease. And so we basically spend our time studying mutations. So we have a lot of information about mutations. And what we see is they're negative. I've never seen a positive mutation in the thousands that we've looked at in cancer research. They're invariably negative.
So when you see mutations, it's a disease, it's a problem, it's something bad, not something good.
Almost always right. There are a few positive ones, but we understand those. They're not a problem for creationists. But by and large, what we see is 99.99% are clearly negative.
Now, this is a big question, but that's all right. You've done a lot of study. How many positive mutations would it take to get from that goo to you?
Well, people have estimated, it's obviously a guess, one doesn't know.
Sure, I mean, we're talking here in speculation.
But those people who have speculated on this say it takes billions, if not many, many billions. So we're talking about a huge, huge number, and of course if 99.99% are negative,
they're going to cancel out those couple positives that happen.
Right, which is exactly what happens.
And we begin to say that, this whole idea that the whole thing could have happened by evolution, by mutation, it just isn't possible.
From what we understand now, yes, it's certainly beyond the realm of what we understand probability to be.
My whole life I've listened to people and they've equated evolution with science, and they've equated creation with religion. And they've said that the scientific world sees this whole thing from a perspective of evolution, and us creationists, we're just these radical religionists who don't look at science. But today you're telling me that science is on the side of creation, and good science simply isn't proving evolution.
Yeah, I know many Christians who became Christians because of their study of science.
Isn't that amazing? My friend, I want to speak to you for a moment, and I just want you to know that we've got scientist after scientist telling us that when we really put evolution under the microscope, when we look at the evidence that they're giving us, it isn't a creation versus evolution debate. Evolution isn't possible. It just doesn't work. We always come back to the reality that our origin is in God, our Creator. And today we're talking about mutations, which ties in the whole exciting scientific study of DNA with this whole business of evolution, because this is where the mutations would have to take place for evolution to happen.
Right. The basis of evolution is clearly mutations. So that's the foundation for the theory.
And mutations have to take place in the DNA chain.
Right. That's the source of the material which natural selection selects from is mutation. That's the ultimate source.
Okay. Now, today we're going to, we've got on the screen there, mutations prove creation. What does that mean? Why do you say that?
Well, because when we study mutations, we find, and I looked at quite a few mutations, and the question is, are beneficial mutations common? That's because of course, since evolution is based on mutations, are there many of them? Are they common?
And it would take positive mutation, not negative mutation, for life to advance that way.
Right. What they call beneficial or favorable. They sometimes use the term favorable. Okay. So I did a literature research. I looked at the scientific literature, and this is what I found. And I found a total of 170,000 mutations in a database called Biological Abstracts. And I found in Medline, another database, 283,205 mutations. This is all mutations, period. Of those that were beneficial, I found 98 in Biological Abstracts, 88 in Medline, a total of 186 beneficial mutations I located in the literature.
Out of almost 500,000.
Right.
Okay.
Now, some of these were articles that talked about the same mutation, so it's actually not a total of 186 beneficial mutations.
Oh, those are just articles.
Those are just articles, and many talked about the same mutations.
Okay.
And there's some overlap between Biological Abstracts and Medline. They abstract some of the same journals. So this information isn't quite correct, but it gives you an idea of the small percent of the mutations described in the literature, the small percent that are beneficial, which turns out to be about 0.04%, which is considerably less than 1% of the mutations that's described in the literature were beneficial.
That's amazing.
So it's a very, very, very small number,
Tiny,
as
tiny
you can
amount.
see. And of course, as we do more research, this will undoubtedly change. But my guess is it's not going to change too much one way or the other.
It's probably not going to go up, is it?
I doubt it. If it does, if it goes up, it may go up slightly, but that's all. And some examples of beneficial mutations. And I'm in the process now of looking at these, trying to find out, what mutations occurred, what changes occurred to make them beneficial. And I found that many were beneficial only for humans. They were not beneficial for the plant or the animal. And the best example is seedless fruit. That's a result of mutation, which is convenient for us. We can eat our watermelon without having to worry about seeds, or our grapefruit without having to worry about seeds. But it's certainly not beneficial for in this case, the plant.
So even when we're talking about beneficial mutations, we have to say beneficial for whom?
For whom, right.
That's right.
An important consideration.
And by the way, I love seedless watermelons. I think that's the way to go.
Yeah, well, it's...
Unless you're sitting in the yard, then it's all right to spit, but not at the table. So this is a really good deal for us. But if I were a watermelon or a grapefruit, I wouldn't be excited about not having any successors coming because I had no seeds.
Yeah, reproduction is a little problem for these plants. That's true. And another example is they found in what they call Belgian blue cattle. There's a muscle mutation which produces beef which is more valuable to farmers because they end up with more muscle, they end up with more meat, they end up with more profit. So it's clearly beneficial to the farmer, and they're also low in fat. So it's tender, it's low in fat, so it's ideal for the farmer.
So even when we're talking about mutations being beneficial, depends on what point of view you're looking at.
Right, if we're the cattle in this case, or you're the farmer. And now let's look at the cattle. Well, the side effects for the cattle include a reduction of the animal's fertility. So it's not as fertile. So from the cattle standpoint, it's certainly not, again, a beneficial mutation. And another example, this is my favorite because I did a whole article on this. In 1791, Seth Wright discovered one of the sheep in his flock had really short legs. It resembled actually a dachshund. And he thought, now wait a minute, these sheep don't jump very high. They don't jump fences. So I can save money by having very low cost, low fences, and they won't jump the fences. Boy, this is an ideal mutation for a sheep herder because it save a lot of money. And as far as they could see, the meat was just as good, the fur was just as good, the wool. So they thought, gee, this is great. But what they found was that this is actually a lethal deformity which is called achondroplasia. And this is a normal sheep, but the mutation basically caused the legs to be not this long but much, much shorter. So as a result, it just looked like a normal sheep with very short legs.
And I'm reading ahead of you there, but that led finally to the extinction of that species of sheep. Which when you put that into the whole evolutionary thing, again, the mutations produce elimination. They don't produce a stronger, moving up the scale species.
Right. It was a mutation basically in the cartilage production. So the cartilage was deficient. So as a result, the cartilage, not only in the legs were a problem, but the cartilage elsewhere in the body were a problem. And they took a lot of these and did autopsies on them and studied them. In fact, there are a number of journal articles where they autopsied and carefully studied these sheep, and they found they had a lot of structural defects. So it was a major mutation. So we can see it's not indeed a beneficial result, it was a negative result. And it's interesting, this example was one of the prime examples Darwinists used to prove what they called mega mutations, which you have one mutation and you have a whole new breed. Because they actually called this a breed. It was called the Ancon breed of sheep. And they thought, well, gee, if we can get this whole new breed with one mutation, maybe we can get a drastic change in life with one mutation. We can have a mega step forward in evolution. And Darwin talked about this. Many of his contemporaries talked about this. So this was an exciting step that Darwin saw in evolution. Of course, it turned out to be
a step backwards, not a step forwards.
Totally a step backwards, right. Exactly what happened. And another example, this is an example of a beneficial mutation which is talked about a lot today in the literature. And this is a mutation which causes bacteria to be resistant to antibiotics. And this is a good illustration of what happens, how bacteria become resistant to antibiotics. What happens here is in the ribosome, and the ribosome is a structure inside of the cell, and the ribosome, this is the normal, picturing the normal structure. And a mutation basically prevents this normal fit. This is streptomycin. Streptomycin fits in here, and as a result, we can see it blocks what this section here does. So it's like a key in a lock. If the key fits, you can get in the door. If the key doesn't fit, you can't get in the door. So in order for streptomycin to work, the key has to fit. That's how it works, by the fit here. Well, a mutation, you can see, causes a little notch here because the shape now of the protein is different. So as a result, the streptomycin doesn't fit. So as a result, this bacteria now is resistant against streptomycin. It no longer is killed by streptomycin. So this bacteria clearly has an advantage in an environment with streptomycin. The problem is, though, in the wild, this does not have an advantage. It has a clear disadvantage. And the example I use in my class is that you go through a divorce, and your husband has a key to the house, and he can get in and take your things. How do you keep him out? Well, you change the locks, but of course can't do that here.
Right.
So the illustration here is you put some gum inside the locks,
Okay.
and he can't get in the house anymore. So you kept your husband out. The problem is you can't get in either. So it achieves the goal of keeping him out, but it also prevents you from getting in. But on the other hand, if you have a back door and maybe another key for that, you can still use the house. Well, likewise, the bacteria, they can still survive in spite of this mutation. But in an environment where you have streptomycin, they have a clear advantage. But they don't have an advantage in the wild.
So it isn't making it stronger or better, it just makes it resistant to that one thing.
Right. And
Okay.
in an environment which has the antibiotic. And when the patient goes home, even if they have resistant bacteria, sometimes back in the normal environment where they have the resistant bacteria compete with the normal bacteria, the resistant bacteria often cannot compete, and they often die out. And that's called a fitness cost. And
Fitness cost.
fitness cost. And all mutations that I know of in bacteria which confer resistance against an antibiotic have a fitness cost. And there are many other examples. And one which I think is really fascinating is bacteria have pumps, where the pump basically pumps out of the bacteria, it pumps out the antibiotic. So you're giving antibiotics to the patient, it's being taken in by the bacteria, and the bacteria is pumping it out.
Huh.
Well, every now and then you have a bacteria that has a broken pump, except it being broken so it doesn't work because those bacteria would die. It's broken so it's on high. It's kind of jammed in high gear. It's kind of like you put your accelerator on your car to the floor and it's jammed. So your engine is running in high speed and you can't stop it. Well, that's what happens here. So what happens is when these pumps are jammed on high, it pumps this stuff out really, really quick. And therefore, it has some resistance against the antibiotic. But in a normal environment, this pump is jammed on high, it uses too much energy, and as a result, that bacteria does not have an advantage in a normal antibiotic-free environment. So again, it's beneficial only in a situation where you have the antibiotic. It's not beneficial in a normal antibiotic-free environment.
So would it be fair to say, before you jump to the next example, would it be fair to say that the positive mutations are just adaptive, but they are not progressive?
Right. They're adaptive in a very limited situation.
But they are not moving it on to become something bigger or better.
Right. They're not, as they call these superbacteria, but they're not. They're not more evolved bacteria.
Yeah, that's interesting.
And the last example I'll use is resistance to AIDS virus. And this is the AIDS virus here. And the AIDS virus, to get in the cell, has to have, in essence, two keys. Again, the lock and the key example. And the keys are basically receptors. The CD4 is one and the CCR5 is another. And to get in, they've got to have the keys to both of those receptors. Well, the AIDS virus has that. But now there are some cases where you have a mutation where this receptor here, and this receptor is made inside of the cell and is brought up to the membrane and then installed in the membrane. These receptors are damaged because of mutation. Therefore, this receptor is partially constructed. It never makes it to the surface. Therefore, this bacteria doesn't have the other receptor. Therefore, the AIDS virus cannot get into the cell. Therefore, this patient is largely resistant to the AIDS virus. They can be exposed to lots of AIDS virus, and they do just fine. They have no problem. But they are not evolving. These people are not more evolved. They have damage which this receptor doesn't work, and as a result, they have an advantage in a situation where they have the HIV virus. In other situation, keep in mind this receptor doesn't work. So what this receptor doesn't take place because it's broken. And therefore it has, again, a fitness cost in a normal environment.
So we're just not seeing mutations move us towards becoming stronger within a species, much less move from species to species.
Right. What evolution would require is evolving a whole new receptor, which is a new structure which functions to keep the HIV out. In that case, then it would be evolving forward, which is what Darwinism requires. But we don't see that, at least in all the examples that I've looked at, we don't see that. Now, I'm not saying examples don't exist. Certainly in the millions of mutations that exist, there's bound to be one that's clearly beneficial. I'm not saying they don't exist. I'm saying they're really extremely rare.
Very, very, very rare. Have you seen one in all your study?
I've never seen one in my work, no.
All of your study, the mutation has either been negative, which leads to the death, or it's been adaptive, but not really progressive.
Right. If I discovered one, I would be very rich and famous.
Well, if there's one out there, I hope you discover it.
Yeah, I'd sure like to.
But with evolution, we're playing a numbers game, aren't we?
Right.
And so, so we're going to talk about the number of mutations required for Darwin's theory to work. But before we do that, we've got to take a break. So don't you go away. You come back. We're going to see if this is possible in any realm. I'm going to give you a hint it isn't, but you come back and be with us anyhow.
Creation versus evolution. You weigh the evidence. Skunk cabbage. Amazing abilities. The skunk cabbage is a uniquely designed plant which also has a built-in thermostat. If the air temperature drops as low as 10 degrees Fahrenheit, the skunk cabbage produces the heat it needs to maintain a temperature between 72 and 74 degrees. If the flower becomes too warm, heat is removed. Because of its amazing abilities, it is the first plant to break through the snow in early spring. How could this intricately designed flower be the result of random chance processes?
Hi, we're back with Dr. Jerry Bergman. We're talking about mutations, and we're talking about positive mutations. We're finding out that word positive is relative to from whose perspective you're looking at it, if I'm right. Before we go to the number of mutations required, let's go back to this HIV cell and talk about how it becomes resistant. We'll use the DNA model over here and show us how does that happen.
Yeah, basically, in the case of the resistance to HIV, a receptor is not formed properly. And the reason it's not formed properly is because this code here, this is the code that produces the protein. One of the letters on this code is changed. We have a different letter. And as a result, the protein code, the chain which is produced, is not produced properly. So as a result, it doesn't fold properly because you have the wrong letter in there. So because it doesn't fold properly, it doesn't work. And that means when it's carried to the cell membrane to be put in the cell membrane to work as a receptor, it doesn't work because it's defective. It would be like in typing a sentence if you misspell a word. It doesn't make sense beyond that point. Now, we can often read words that are misspelled. We can figure out what it's supposed to say. But in this case, it can't. If you have the wrong letter in there, and that letter produces a code which doesn't fold properly, doesn't result in proper folding, it's not going to work. And as a result, that receptor does not function. And if that receptor is not there, that HIV virus cannot get into the cell. It's like to get into a house, you have to have two keys. The HIV to get into the cell, it's got to have both keys. They both have to fit. In this case, the virus is okay. The receptor, the lock, key in a lock example again, the lock is broken, it doesn't work, therefore the HIV virus can't get into the cell. Therefore, it cannot infect the patient. Therefore, the patient cannot get AIDS.
But that isn't making it a better cell. It's really a deformity.
Right. It's a deformity, and of course it's not an advantage except in this one particular situation.
So it's not really a positive mutation. I mean, it's a positive thing that it's resistant to HIV, but it's not positive in the sense that it's making it a stronger, better cell. Is that right?
Right. It's positive in a very limited situation.
That's right.
But assuming now we do have a positive mutation, we'll call it a positive mutation.
We haven't found one yet, but if you find one, let's assume we did. Okay. Now let's talk about the numbers. Okay.
And so how many do we need? And now this again is estimating. People are guesstimating actually, but some people have tried to guesstimate how many we need. And one person estimated about 1,000 to 2,000 different proteins are used in our body, okay?
This is just the human body.
The human body.
Okay.
So we have 1,000 to 2,000 proteins. Different proteins are used. Now some of these are very similar. They're different forms of the same protein type. But we'll assume for the sake of argument, we'll assume we've got 1,000. Okay, and these are all produced by the 35,000 human genes that Darwinism teaches evolved from other, less complex, often smaller genes. Okay? So we've got 1,000 to 2,000 different proteins. We've got 35,000 genes. So basically what we have to do is evolve these 35,000 human genes. Now, what's the likelihood of doing that? How many mutations do we need? Well, Shermer, he estimates that trillions of distinct modifications, i.e. mutations, were required to evolve humans alone. Now, Shermer, by the way, is an atheist. He's not a Christian. He's not a believer. So he estimates that trillions are required. And clearly, billions are required. How many billions, we don't know, but he estimates trillions are required. Each modification, then, probably would require many mutations, because we can get a mutation which produces a beneficial change. Then we get another mutation which goes backward. So to get the set of mutations we need, we have to have many more mutations than is required, because it's like going 2 feet up and 1 foot down. We progress and then we go backward, and we don't end up in the same position, but we still go backward. Okay? The fact is, though, beneficial mutations are very rare and virtually always carry a fitness cost, as we talked about earlier. So they're rare, number one. So how many, if we need trillions required to evolve us, how many do we need, period? We need many, 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 many trillions of mutations. And so I conclude from this, okay, that other mechanisms, even though they've been proposed, at its core, macroevolution requires new information. And the ultimate source of new information is mutations. And my conclusion is mutations simply cannot provide the source of genetic variation we need. And looking at that from many angles, we see we just don't get what we need to produce what we have.
So, does the word impossible come into play here, Dr. Jerry?
I would say the best word that describes the situation right is it's realistically impossible.
It becomes statistically impossible for this to work.
Right. Now, Darwinists say, yes, it's improbable, but we're here.
Yes.
We can't use a God explanation, because that's theology. We can't resort to that explanation. So, yes, it's highly improbable, but improbable things happen. Here we are. We know we're here.
So it only becomes likely when it's like saying, we know the answer's over here, but we're not going to look over there. So as long as we're looking over here, we have to assume this to be true.
Right. They assume this is true in spite of the evidence.
Because they've already eliminated God as a possibility.
Right.
So who's being objective here? It seems to me that if you're saying God could have made this or it could have evolved, you're going to very obviously quickly come to the point where you're going to say God made it, because the other side is improbable to the point of impossible.
Right. They say the rules of science are such that you cannot appeal to God. That's what they say. But my question is, who made those rules up?
Yeah, they made the rules.
And don't those rules limit? Why limit explanations?
You know, it seems to me, whenever I hear the term mutation, it takes me from the front page of the paper that's talking about facts and history and what's going on in current events in the world to the comic page. I mean, when I enter the realm of science fiction and I begin to pretend that we can have a Spider-Man and a Ninja Turtle and a Superman because these things have happened, that's fun for the comic page. But when we enter the world of reality, mutations simply do not make us better.
Right.
And so it seems to me that this is all a fairy tale. It's science fiction. It's not science. And I think that is the point that we come to. We cannot eliminate God. God is there, and God has made us.
Right.
My friend, I just want to wrap this up today by talking to you about the fact that it's God's view that He made you. That's what we say at the end of every show. And the people who are coming up with these theories are people who have said, we must eliminate the truth so that we can look for other answers. I don't believe you're in that camp. I believe that you're a person who will look around, and as the Scriptures say, that creation declares the glory of God. And as long as you intuitively know that, I believe you're searching for truth with us. My friends, I want to tell you, God has made us, and the evidence is overwhelming. And now we're beginning to see that the evidence of science refutes, doesn't confirm, it refutes the theory of evolution. And so I just want to remind you as we leave today, I want to thank Dr. Jerry. It's been a great show.
Thank you.
You've given us awesome evidence. But I just want to remind our viewers as we go today that it's still God's view that He created you. And that should be your worldview too. Till next time, God bless you, and I hope to see you again.